Sorry for delay. And he received a postdoc fellowship Humboldt from Germany. He was visited as visiting scientist and for many training and research program to the many countries continental like Minnesota, USA, Sri Lanka, Quetta, Japan, London, UK. He's having teaching experience at about 34 years and research at about 21 years. Professor Guru Subramanian sir is having 11 awards and recognition. Research grant receiver from DST, New Delhi, Indo USA, DBT, SERB, DST, New Delhi, and many others awards are is recognized. Research paper is having in her at about 132 national as well as international. Along the six books, book chapter 19 and his guided PhD 19 awarded student and MPhil 11 awarded student. So respected sir, we welcome you all. Again, dear participants, I would like to mention that after such enlightening on said topic, this platform will be open to you for questions and queries. So thank you again, sir. We can proceed or start this webinar.
Thank you so much, sir. Thank you so much for your
Uh,
nice introduction. And
Thank you,
before
sir. Thank you,
going
sir.
into my speech, I would like to thank Professor K.R.S. Sambasivarao for giving me an opportunity to share my research experience in next generation sequencing. And I would like to thank the organizers arranging this kind of very good online platform, which is very useful to the student community as well as the research community. Because Professor K.R.S. Sambasivarao is very famous in all over the world as well as in India because of during COVID time, he conducted around 2,500 webinar courses. So that is a real achievement from his end. So with this introduction, with the due permission, I would like to share my slide before this August
You just open
presents.
your PPT and then go for sharing, sir.
Okay.
You just open your PPT at desktop, and then there is arrow mark. No? You can go to arrow mark and then open, sir. Yes, we are able to see your presentation.
This in full screen, sir.
Full screen, I don't know whether it is full screen. You just try for full screen. No, it is gone. It is gone, sir. Presentation is removed. You just go re-upload. No, you just, sir, you open your presentation first on desktop and then start
Yes, sir.
sharing, sir.
Okay, sir. I have done, sir.
Yes,
Now
it
it
is
is
visible,
coming.
sir. Visible, sir.
Is it full screen?
It is not a full screen, sir, but it is visible.
It is visible.
Yes, sir. But I think in full screen is getting problem.
Sir, you go ahead with this, sir. Let us see if possible.
Okay. Today I'm going to discuss about next generation sequencing and its applications. So normally you know that nowadays a lot of research groups working on this next generation sequencing around the world. So what is this here?
Any problem, sir?
Yeah. Yes, sir.
The presentation has gone, sir.
That's why, sir. That I'm not in a position to move this slide.
Oh, okay.
Convert in. If you are facing problems, or okay.
Slides are now not moving.
Just move it. Now you are Yutran again.
No, sir, the slide are not moving.
Can you, otherwise you send to here? I will try.
Okay, sir.
You just move it before you upload, share the slides, sir. Then if it is moving, I think it should go.
I have sent it to you, sir.
Yes, sir. Yes, sir. I'm just opening.
Okay, sir. Yeah, later. Sorry.
So are you able to see, sir?
Yeah. Now only, yes, sir. It's opening.
It is coming now?
Yes, sir.
Yeah.
It's coming. It's visible also,
Yes,
sir.
sir. Yes, sir. Yes, sir.
You can make it to full screen, sir.
I'm just trying.
Okay, okay.
Kishore? Kishore, bola. Full screen. It is not. It is also not becoming full screen, sir, for me.
That's problem is there, sir.
Full screen.
But whether it is moving, sir, the
Full
slide
screen,
is.
sir. Full screen. Just 1 second.
Yeah.
Please. Sir, it has come?
No, sir.
Huh?
No,
No, sir, it is
previously
not
it
visible,
was
sir.
there, but yes, make it full
Now it
screen.
is came.
It has
Yeah,
come,
now
sir?
it's
Yes, sir.
clear,
Now,
sir.
Yes,
Yeah.
sir. Full screen.
But slide is
Full
moving,
screen,
sir. Next slide, please.
It's moving, sir. It is moving. Kana?
no, not moving,
It
sir.
is
Not
moving, no?
No, no,
Not
not
It
moving,
is
sir.
moving. It is moving
Yes,
on
yes.
our
Yes,
side.
yes, yes.
No,
Yeah,
no,
yeah,
no.
correct, sir. Yes,
Yes,
yes.
yes,
it's
sir.
moving, sir.
Yes, sir. Yes, sir. You can
Okay,
go ahead, sir.
move to next slide, sir. Next slide, sir.
Yes, sir.
See, now we are talking about DNA sequencing. Whether DNA, RNA, or protein, whatever sequencing, whenever you are designating the term sequencing, means we are ordering that either DNA or RNA or amino acid molecules. Please remember that point. That is the meaning of sequencing. So the father of sequencing is Sanger, because we started with Sanger sequencing. Next slide, sir.
Yes, sir.
So in that, this is your first generation sequencing. In this thing, the speciality of this Sanger sequencing is normally we are using primer, DNA, DTNTP, everything, DNA polymerase, everything we are using. But here we are adding one more thing that is called that is called a DDNTP, dideoxy-NTP. So in that part, our OH molecule is missing. So normally we are mixing in terms of 1:100. So whenever this dideoxy-NTP molecule is coming into that, it will terminate the chain. That's why Sanger sequencing is otherwise known as chain termination method. This is your first part. Previously, we did everything in terms of radio-labeled nucleotides. And nowadays, we are going for automated Sanger sequencing. In that part, to detect nucleotides, we are using 4 different types of fluoropores. So that way, easily the machine will identify what kind of base pair it is. So Sanger sequencing is nothing but the basic principle behind is capillary electrophoresis only. So here the basic objective is whenever that dideoxy NTP is entering, then your chain formation will stop. The next slide, please, sir. So in that Sanger sequencing, very important point is, first we are fragmenting the DNA. Then we have cloned with the plasmid vector, then separate by electrophoresis. Then we are reading out in terms of fluorescent tags. Next slide, sir. Now, if you are coming to this first generation sequencing, actually speaking, Sanger chain termination method and Maxim Gilbert sequencing method. Same year, one is from UK, Sanger sequencing, and Maxim Gilbert sequencing from USA. But most of the scientists, they have well used with respect to only Sanger sequencing, because Maxim Gilbert sequencing, there is a lot of radio-labeled nucleotides involved. So because of that, that particular sequencing method is not that much popular. And it is very cumbersome also. So that is why, with respect to our sequencing part, our Sanger sequencing is the gold standard method. Next slide, please. So now if you're coming to next generation sequencing or high throughput sequencing, all are same. So in this thing, next generation sequencing and your Sanger sequencing. In Sanger sequencing, you can read one molecule per sample. But whereas in next generation sequencing, you can read millions of reads per samples. So that is the basic difference between your Sanger sequencing and next generation sequencing. Next slide. So now if you are seeing that history of DNA sequencing, especially next generation sequencing. Next generation sequencing really started in the year 2006 only. Yes. Next slide. So already I have informed about what are all the differences between Sanger sequencing and next generation sequencing. Because here, very important point is in next generation sequencing, library preparation. That is playing a major role in next generation sequencing. And normally in Sanger sequencing, we are using 96 well plate or 384 well plate. But in terms of next generation sequencing, we are using only the flow cell. Flow cell, otherwise known as, it looks like slide, our normal microscopic slide, flow cell. So that flow cell also, lane also, it is coming around 1 lane to 16 lanes are there. Then another important point is in Sanger sequencing, yes, 1 read per sample. But whereas in NGS, millions of reads per sample. That is a basic difference between Sanger sequencing and next generation sequencing. Next slide, please. Yeah. So now, high throughput sequencing, next generation sequencing, otherwise also known as massive parallel sequencing. So massive means enormous amount of data we can collect. That is number one. Parallelly, more than, yes, even in Sanger sequencing, we are handling only one sample. But here, yes, we can parallelly, we can get so many millions of reads we make, or data we can collect it by using this particular technology. That is why it is known as massive parallel sequencing method. Next slide. Yes. So now you may ask a question. Sir, Sanger sequencing is a gold standard and reliability is more. But next generation sequencing, yes, it is not with respect to sensitivity and specificity. Yes, it's not that much equal to your Sanger sequencing. Yes, that's why we have validated so many platforms by which here the main aim of that next generation sequencing is, one is reduce the sample size. That is number one. And number two, reduce the cost. And within minimum time, less time, yes, we can sequence millions of samples. So these are all the advantages with respect to next generation sequencing. Next slide. Yeah, now if you're coming to the next generation sequencing, I already told it is started from the year 2006 only. Because in the year 2005, first company called 454 Roche, yeah, they have started that emulsion PCR technology. Yes, that is one platform you should know about. I hope you know already that part. 454 Roche, that is one thing. Then in 2006 only, this Illumina sequencing or Solexa sequencing came into existence. And the same year 2006, yes, another one more platform that is called Solid. So these are all NGS platforms. 454 Roche pyro sequencing, that is in the year 2005. And in 2006, two things, one is Illumina and another one is Solid. So Solid thing by Applied Biosystems. Then later in 2010 only, yes, a lot of developments with respect to your Illumina as well as Solid system has come. So in 2011 only, another one more platform that is known as Ion Torrent as well as PacBio. So there are four platforms are there. Roche 454 pyro sequencing, Illumina sequencing, Solid sequencing, Ion Torrent sequencing, and PacBio sequencing. These are all the different kind of platforms. So that way, yes, we are getting millions of data with less time period. Next slide, please. Okay. I have already mentioned about in this thing. So in this next generation sequencing, yeah, there are 5 things are there. 454 Roche, Illumina, Solid, Ion Torrent, and PacBio. So here very important point is, one is how you are amplifying your target. That is number one. And what kind of chemistry and sequence image technology you are following. This way, little bit variations are there. So you should know about, yeah, there are around 5 different types of NGS platforms. In that thing, yeah, how you are amplifying the target. One is your emulsion PCR. That is called clonal amplification. Everything is clonal amplification only, but technology little bit different. In 454 Roche, we are following emulsion PCR. But whereas in Illumina, that is known as bridge amplification, bridge PCR. Yes. So, and whenever you are coming to that Ion Torrent, yeah, on-bead emulsion PCR, little bit differences are there. And with respect to that technology wise, if you are seeing there, that means sequencing chemistry and imaging technology. If you are considering your 454 Roche, that particular platform following bioluminescent technology. Bioluminescent technology by which we are identifying our nucleotides. That is one part. That is 454 Roche sequencing platform. But in Illumina sequencing, yes, here we are using reversible terminator technology. But here we are using in 454 Roche platform, we are using beads. But whereas in Illumina sequencing, we are using flow cell. And we are another technology known as reversible terminator technology. So that is why Illumina is very famous. Most of the research groups working on using only Illumina platform only. Then Another thing is, whenever you are coming to that ion torrent sequencing platform, yes, their main sequencing chemistry and imaging technology, if you are coming here, one is they are detecting the nucleotides based upon hydrogen ion concentration. And how we are imaging by using chip. Yes, semiconductor chip sequencing. So these are all the overall description about the next generation sequencing platforms, but we will see in detail one by one. Next slide, sir. Yes. So if you are seeing overall methodology, whatever sequencing platform you are using, yes, 3 steps are there. First one is your library preparation. That is number 1, first step. And second step is your sequencing and imaging. And third one is your data analysis. So whatever NGS platform the research group using, yes, the basic principle is only these 3 things: library preparation, sequencing and imaging, and your bioinformatic data analysis. That's why here, whenever you are working on next generation sequencing, there are a lot of bioinformatic pipelines are there. So for this, very important point is you should have basic knowledge about Linux system and R language. So these 2 things is fundamental things you should know. You are very good in R language and a Linux based system you are using. Yes, then only you can analyze your data in different bioinformatic pipelines, and you can present your data. Next slide, please. So now if you are coming to here, this library preparation. In library preparation, really what we are doing? See, in library preparation, one is first your DNA template. Yes, we are constructing, and after that we are amplifying by using PCR. So that is very, very important part. Already you know that your nucleic acid is, yes, it's a macromolecule. So you fragment all the DNA into, yes, small nucleotides. Yes, then, yes, we are constructing that library. Once your library is constructed, then we are amplifying. Next slide, please. And in sequencing and imaging, yeah, here it is very, very important part, because already in Sanger sequencing, we are using 4 nucleotide, 4 nucleobases with different fluorophore you are using, different color you are using. So by using that machine, based upon that color, fluorescent color, machine is identifying that particular nucleobases. But here, what is happening here? Yeah, here we are doing sequencing. And another important point is image, we are doing single nucleobases. Each and every nucleobases, yes, everything is imaged. Then, yes, one by one, machine will read and it will give all the informations. So that is very, very important part. So one cycle means, yeah, there are 4 nucleobases are there. Yes. Next slide, please. And the data analysis, I have already told, so many is freely available, open sources are there. Only thing is, you have to have knowledge about how to utilize the bioinformatic tool for your utility. That is very, very important thing. So normally, see, we are thinking that because normally we are outsourcing the sequencing. All next generation sequencing, we are outsourcing. Yes, but you are not good at data analysis. So what kind of tool you have to use, how you are going to present your data, and how you are going to publish your data. in the high impact factor journal or Q1 journal. Yes, that point, yeah, you have to have basic knowledge about everything you should know. Otherwise, very, very difficult. If you are depending only on that particular outsourcing company, whatever, because they are having a lot of work. So they will not individually, they will not give any attention to what we are in need of. Next slide, please. Yeah. So now if you're seeing here, that's why I told widely adapted and next generation sequencing. If you are seeing here, yes, one is very, very important point is Roche diagnostics. So protocol is pyrosequencing, 454 pyrosequencing. Then Illumina, yes, is Solaxa sequencing. So there they are following bridge amplification, bridge PCR, and reversible terminator technology. That is Solaxa sequencing. Then if you are coming to that life technology, that is solid. So by using ligation, yes, we are detecting the nucleobases. Then your single heliscope, single molecule sequencing. Yeah, that is Helices Biosciences. So many companies, that means companies means so many platforms are there. So according to your need, what kind of platform you are going to use, that thing you have to decide, because everything is money involved. Because normally if you see, whenever you are going for metagenome analysis or transcriptome analysis, whenever you are going for metagenome analysis, so normally, yeah, per sample, they are charging around 10,000 rupees. 10,000 rupees they are charging. And whenever you are going for shotgun sequencing, that means if you want to know the functional aspects of that particular subject, then it will take 40,000 rupees. This is, I am telling you about metagenomic things. That means 16S rRNA sequencing. But whenever you are coming to the transcriptomics level, if you are coming here, yes, per sample, they are charging 25,000 rupees. So that is why here, suppose if you are using Illumina platform, yes, because don't commit any package. You have to ask that particular outsourcing person, you have to book only the lane size. You have to book the lane only. So that way your cost may reduce. Because this thing they may not tell. They will say, okay, sir, as a whole. And another important point is if you are very good in bioinformatic part, then don't include bioinformatic analysis, because we can do all the analysis by ourselves. Because and lot of free software tools are there. Only thing is you have to spend some time and you have to learn and you have to do it. It will take some time. That's all. So that way we can reduce the cost. Next slide, please. So whenever you are coming to that applications as a whole, if you're seeing there, yes, in whatever field you are working, yeah, everywhere now sequencing playing a major role. Whatever it is, whether it is a transcriptomic study or metagenomic study or whole genome sequencing or whole exome sequencing, everywhere, yes, whatever may be your field, you can utilize this next generation sequencing platforms. Next slide, please. Now, if you're coming to that workflow, that's why I told there are around 5 NGS platforms are there. 454 pyrosequencing, Solid, Illumina, PacBio, Ion Torrent. If you are seeing here, yes, already I told here. One is library preparation. In that library preparation, really what we are doing. See, first you have to extract your DNA or RNA from the sample. That is number one. Then you have to do the quality test. That is number two. Once it is quality is passed, yeah, then only you can further, you can go for other thing. Because if your quality you are compromising, then there is a lot of problem with the data. Please remember this point, note this point. If your sample, either DNA or RNA, is not following or not passing your quality test, don't go for. Clearly you have to tell that outsourcing person that yes, it is not passing. They may say, sir, no problem, sir, you may get the data. No, whether it is a DNA and RNA, quality check is very, very essential. If it is passing the test, then only you have to tell go for sequencing. So once that pass that technology, after that what they are doing, they are fragmenting the DNA. Then after that, see, your DNA is fragmented. Yes, how I know about what kind of fragments are there? It's very difficult. So that is why what we are doing, yes, we are adding adapter. We are adding adapter. So see this brown color, whatever color it is there, those are all your DNA sequence. And either 3' end or at 5' end, everywhere you are adding adapter containing your barcode. So with the help of that barcode, you can identify each and every DNA fragment. Please note this point. Very, very important thing. In library preparation, you are doing only that point. So in your library preparation, yes, adapter, clearly it has to attach to your DNA fragment. So with the help of that adapter only, yes, we are going to do all other job. Okay, this is library preparation. Then if you are coming to two things are there. One is emulsion PCR, another is bridge PCR. Two things are there. So your Illumina part, yes, going for bridge PCR. But whereas your 454 pyro sequencing or solid platform, all they are going for emulsion PCR, ion torrent, all the three platforms going for emulsion PCR. Whenever you are seeing emulsion PCR, that is nothing but oil. See, beads are there. Your beads, yes, beads containing your DNA fragments. Okay, beads, this is nothing, otherwise it is known as bead technology. So in oil droplet, yes, this, that is why it is known as emulsion PCR. We are doing that one. So that kind of amplification followed in pyro sequencing as well as the solid NGS platform. Please remember that thing. Whereas in Illumina sequencing, yes, we are following bridge PCR. Please remember that thing. Everywhere clonal amplification is there. That is the speciality of next generation sequencing. Please remember, that is the basic difference between your normal PCR. In Sanger sequencing, normal PCR you are using. That is not clonal amplification. That is only single thing. But whereas whenever you are coming to NGS, here we are using clonal amplification. Either we are following emulsion PCR or bridge PCR. So this way a little bit differences are there in your NGS platforms. Next slide. Yes. Then already I told in next generation sequencing, yes, especially if you are following Illumina, we are using flow cell. Please remember that thing, because worldwide, yes, most of the research groups, they are following only the flow cell only. So that's why this figure always showing fantastic. Yes, you may not get this kind of picture. Flow cell, it's nothing but your microscopic slide, but it's not microscopic slide. Because there, see, whenever you are coming to that, lane is there. Each lane containing two columns. That's why here you are clearly you are seeing this one. So each lane containing two columns, and each column containing 50 tiles. Please remember that thing. So there only we are going to load our DNA samples. Please remember that thing. See here how we are loading. So lane is there. Lane is containing 2 columns and each column containing 50 tiles. Yes, that is very, very important part. So one flow cell normally containing 8 lanes. So 8 lanes means 8 into 2. So 16 columns are there. Please remember that point. Okay. So in Illumina, we are using flow cell. Okay. Next slide, please. See, this is the different, this is the history of your Illumina thing. Yeah. Normally it is containing 8 channels. The dimension is 75 millimeter into 25 millimeter. Channel depth is 70 micrometer. And channel length is 70 millimeter. And see, we are going to do imaging. That is 60 millimeter. So the last one is your latest part. Yes. So this way, this is our flow cell over the period of time. They have reduced all the things. Next slide, please. Yeah. So now if you're coming to, that's why I told, because whenever you are coming to the next generation sequencing, you should know that basic principle, because different kind of platforms I told, Roche, 454 pyro sequencing, Solid, Illumina, Ion Torrent, PacBio. Yes, here how you are preparing your template, library preparation. That way, little bit difference is there. Then what kind of chemistry you are using? The two thing you should know. That is very, very important part. Whether it is following emulsion PCR or bridge PCR. That is one with respect to library preparation. Then whenever you are coming to chemistry means here imaging technology, because you have to detect your nucleotides. So for that, how they are doing? Yeah, whenever you are coming to Roche, they are using bioluminescence. But whenever you are coming to the Illumina, yes, they are using reversible dye terminator. Same that Sanger sequencing. Same sequencing. So here what they are doing here, that DDNTP, they are adding into that nucleotides. So whenever it comes, yes, every time it will come and it will terminate. It will come, terminate. It will come, terminate. So one cycle is 4 nucleotides. 4 cycles. Please remember that point. Next slide. So this is another platforms. So everywhere you see here, whenever you are coming to the Ion Torrent, yes, technology is, chemistry is your hydrogen ion concentration. Hydrogen ion concentration by which they are deducting the nucleotides. But here also emulsification PCR. But little bit difference is your PacBio. That is single molecule, SMRT. Yes, that is PacBio. Here, what they are doing here, yes, phospho-linked fluorescent nucleotides. Please note, whenever whenever you go to USA, some research groups they are well versed only with PacBio only. Yeah. So PacBio, the basic history chemistry is phospho-linked fluorescent nucleotides. Next slide, please. So now you know about, now you are familiar with what kind of NGS platforms are there. Correct? Roche 454, Illumina, then Solid, PacBio, and Heliscope. So these are all the different kinds of sequencing platform are there. But whatever platform you are using, basic principle is same. Library preparation, then sequencing, imaging, then finally data analysis. Yes. Next. So that is why I'm doing. See here, very important, that's why template preparation. See here, your Roche, Illumina, Solid, these are all clonal amplification. But in Heliscope and PacBio, single molecule sequencing. Please, that's why library preparation, little bit difference is there. Please remember that point. Okay, clonal amplification, PCR amplification is either Roche, Illumina, Solid, all platform, they are following clonal amplification. But whereas your PacBio and Heliscope, they are following single molecule sequencing, they are following. That's why I told chemistry is phospho-linked fluorescent tags. Next slide, please. Yes, that is very, very important thing. And yes, you may ask, is Roche, Solid, and Illumina. With respect to clone, all are doing clonal amplification. Correct. Here, little bit difference is there. Illumina is solid phase bridge PCR. That's why we are using flow cell. Please remember that thing. Illumina bridge PCR. Bridge PCR means we are using flow cell. Solid surface we are using. But whereas your Roche 454 and solid sequencing platform using emulsion PCR. That means your beads containing your DNA as well as the primer that is soaked in the oil droplet. That's why it is known as emulsion PCR. This is the basic difference. Yes, you have to note and you have to be very clear about that basic concepts. Next. Yes. And another important point is, yes, in single molecule sequencing, I already you know about that thing. One is helicose, another is PacBio. Yeah, here we are not going to do any DNA amplification. See, because you want to DNA amplification, you have to use primer. So there lot of error is coming. Whenever you are using primer, error is there. So that is why nowadays we are following primer-free technology. So that is why it is known as single molecule sequencing. That's all. Fragment your DNA. That's all. After that, yes, we are not going to do amplification. Yes, that is very, very important part in PacBio and helicose. And previously, yeah, you see, recently you heard about that Minion sequencing. Yeah, very, very handy type. You can do it at field level. They are getting long reads. But nowadays, what happened? Yes, either in 454 Roche or Illumina and PacBio, yes, we can get longer reads. Only you have to follow that particular protocol. You have to inform that outsourcing thing. We need longer reads. See, small read is there, long read is there. What is the difference between, if your small read means it is within 350 base pair, so your information is very less. But whereas if your read is very long, read means what? Information, data information. So long means, yes, it is more than 400 base pair. Please remember that thing. In PacBio, it is more than 1000 base pair. See, next slide. Yeah. So in that library preparation, yes, already I have told, but here in this library preparation part, one word we are using, that is known as target enrichment. That's all. Target enrichment. So target enrichment means, yes, what is our target? Yes, because your DNA, you are extracting the DNA. Yeah. Then after that, what you are doing? You are fragmenting a whole DNA. Then you are, after that, yes, in that through the target enrichment way by which, yes, you are selecting what kind of target you look into. So that is very, very important. That is the speciality of next generation sequencing. Okay. In library preparation, yes, one important step is out of your DNA fragmentation and adapter ligation. Apart from that, we are doing target enrichment. Yes. Next. Yes. You see, that's why these are all the library preparation. These are all the steps involved. So first step, what you are doing? You are isolating your genomic DNA. Correct. Then after that, what you are doing here? Yeah, we have to identify our, after that, you are fragmenting. After fragmentation, yeah, we have to find out your thing. So that is why you are adding adapter. So that is very, very important thing. Okay. Then after that, you are doing your PCR. So these are all the steps involved in your library preparation. And here we are using in adapter ligation, we are using two terms. One is multiplexing. Multi, yes, more than two. That's all. Multiplexing. Yeah, you are adding, you are mixing so many, yes, sequences. That's all. That is called multiplexing. So that is very, very important part. And you see so many DNA fragments are there. How will you identify? Because based on your barcode. Yes, barcode we are adding. So barcode is nothing but index. So based upon that barcode, yeah, machine will recognize that path. Then it will amplify. Next slide, please. Yeah. So that is why. Next slide, please. See here, very important part is the target enrichment. I told. Yeah, how you are going to maximize your target enrichment. So that is nothing but see target size. What is your size? That is very, very important part. That's why here I have given some example. 50 MB, 5 MB, 500 KB, 50 KB. Yes. What is your desired depth coverage? That is 100. So what is your target size into depth coverage? Always 100 only. Yeah. So that way we are, what is our sequence depth per sample we are calculating. Please remember that point. This is known as target enrichment. So they will ask, you want 5 GB data or this much MB data? What do you want? 6 GB? 10 GB? Yeah. So that way your cost may go high. Please remember that thing. See, if your genome is very small, yeah, we can go for, yes, very small size. Yes. Even if 500 MB or 50 MB or 5 MB, because your genome, whenever you are working on bacteria or virus, very small. But whenever you are working on mammalian, yeah, you need more sequence depth per sample because your target size is high. So you are in need of 10 GB data. Please remember. So you have to familiarize with that particular term. Next slide, please. Yeah. So in this NGS procedure, I already told library preparation. In that library preparation, you are attaching adapter. What is the use of that adapter? We have to, because their barcode is there. Yes, it will identify what kind of fragments are there. And you are doing bridge amplification. So what is that? What is the meaning of bridge amplification? Because see, your adapter containing a particular primer. Yes. So in your flow cell, you are floating your flow cell flooded with a complementary primer of complementary sequence of your adapter primer. Please remember the thing. So then only it will go and then attach. So then once it attached, yes, your DNA polymerase will read that particular bridge. See, what is the meaning of bridge? I will show you that photo. Then you can better way you can understand that thing. And what is the meaning of clonal amplification? Yes, and sequence by synthesis. So this point I will tell you in the coming slides. Next slide, please. Okay. So in that genomic library preparation, you have to fragment your DNA. So normally we are fragment the DNA either by sonication or by tagmentation. So tagmentation is nothing but here, there we are using transposes. See enzymatic method. transposes we are using. Okay. So that is very, very important. So two types of fragmentation is there. One is by sonication, where we are using ultrasonic waves, and segmentation, we are using transposes. That is number one. Right. Then right and left adapter. See, your 3 dash, 5 dash 3 dash and 3 dash 5 dash. Two end is there. So now what we are doing here? Yes, in right thing we are using T7 DNA polymerase. But whereas your left adapter containing T4 DNA ligase. Please note this point. Because yes, two adapter you are using, right adapter, left adapter. Right adapter is containing T7 DNA polymerase and left adapter containing T4 DNA ligase. Yes, that along with your barcode and your DNA template. This is containing, that's why adapter ligation. Then after that, yes. Next slide, please. That's why I told, because normally your adapter, 3 segments, normally it is there. One is your DNA template. Yeah. DNA template. That's why, see, sequence complementary, I told. That means what? In your flow cell, yes. What complementary to that your primer, it is added in your adapter. That thing is there in your flow cell. Please remember sequence complementarity to solid support. That means what thing is there, sequence is there, primer is there in the flow cell. Please remember oligonucleotides on flow cell. Please understand this meaning. And your barcode sequence, that is called index. Index as we are telling. It is normally, it is containing 6 base pairs. Please remember that thing. These are all a standard protocol. 6 bases are there in your barcode. And third one is your binding site. Please. That's why adapter containing 3 portions. One is sequence complementarity to solid support, barcode, and the third one is your binding site. 3 things, because binding site is there, then only your DNA polymerase will go and bind and it will sequence the synthesis. Please remember that part. Okay. So that is very, very important part. Okay. Next slide, please. See, these are all the transposon. These are all the how that this is the description about your adapter. See, this is your barcode, your 5 dash end and 3 dash end. And your adapter, it is there. So this is your blue color is your DNA fragment. Please remember that thing. This way we are attaching each and every fragment we are attaching. Please remember that thing. See here, normally whenever you are sending for sequencing, they will ask, sir, you want a single end or paired end? Yeah, single end means see, you are using only 5 dash 3 dash. So based on 5 dash 3 dash, yes, complementary part we are doing by ourselves. But paired end means, yes, you are reading both the thing. 5 dash 3 dash also, 3 dash 5 dash also. But my suggestion is, yes, go for paired end sequencing. Don't go for, because they will say, sir, that way you can reduce the money. Yeah, but you will not get the proper information. Then whenever you are going for analysis, that time you may face lot of problem. Okay. So always go for paired end sequencing. Next slide, please. Yeah. See how nice way with respect to your DNA fragment, your adapter is attached. Please remember that point. Okay. Next slide, please. See. Yes. See, now your flow cell containing our oligonucleotides complementary to your DNA, whatever it is there in the adapter. So automatically here this flow cell full of your oligonucleotides are there, complementary to that thing. Then once you are loaded, automatically it will go on and bind. Next slide, please. Yeah, bridge amplification. So what is the meaning of bridge amplification? Yes. See here, always you please remember that your right adapter is there, left adapter is there. Correct. Here you know what is the meaning of cluster. Because we are forming one group. Cluster means what? Group. Yes. Whatever here, cluster means your clone. Whatever clone is there, yeah, separate way we are categorizing in that particular flow cell. That is very important part. Next slide, please. See here, this is called bridge amplification. Yeah. You see here, that's why this is your flow cell. See here, how this is containing your oligonucleotide. See, once you loaded your DNA in that tile, please remember that flow cell. 2 column is there, 1 column, whatever that 50 tiles are there. So in that thing, yeah, you load it. Yes, see, automatically it will attach here. Then again, what will do? See, it will form a bridge. So that is why it is known as bridge amplification. So once that bridge is formed, yes, your primer is there. Adapter containing binding site. So automatically what it will do? The primer will attach and it will sequence. So that way it will form same thing. It is forming several things. That is why it is known as cluster. It is forming same copies. See, that's why here clearly they have mentioned as a clonal copies of both forward and reverse strand in a cluster. Clearly, this particular figure shows that thing. So this is happening in your Illumina sequencing, bridge amplification. Bridge amplification, PCR. Next slide. See here, another way of representing your that same bridge amplification. So how that it will go and how that primer will bind to the adapter binding site and it will read. So that way it will form lot of clones as well as a cluster. Next slide. Clonal amplification. That's why it is known as clonal amplification. Please remember that thing. Because once that particular DNA fragment with an adapter, it is bind with your flow cell, automatically what will happen? It will form a bridge. Yes, then after that, your adapter containing binding site. So where your DNA primer will go and then make several copies of clones. Please remember, because in clonal amplification, in Illumina sequencing, playing a major role with respect to quality. I'll show you later. Next slide, please. See here, this is the diagrammatic representation of bridge amplification. See here, how it attached and how it is formed. So that way, you are forming the cluster. I hope now you have understood what is the meaning of bridge amplification and formation of cluster. Cluster is nothing but same copies of that particular DNA fragment. Several copies you are making. Next. Yes. That's why already I told about it. So this kind of bridge PCR amplification we are following in Illumina. Illumina platform, sequencing platform, we are following bridge amplification. Next. Correct. So here, that's why I'm telling you, cluster. So here you know about this thing, because chip is there. So now if you are coming to that imaging technology, yes. So your DNA fragment, yes, with respect to that particular clonal population already there. See here, already clonal populations are there here. So now, yes, you are making as a group cluster. So then you are reading that part with the help of your reversible dye technology. Next slide, please. So this is bridge PCR. It is there in Illumina, correct? But whenever you are coming to that emulsion PCR, yes, here what is happening here? Yes, very important point is here also adapter, everything same, but it is there in your bead. See, one PCR primer is attached to the surface of a bead. So your fragments of your adapter, that means what? Your DNA fragments attached to that adapter. That is called any DNA fragment attached to the adapter. We are telling library. Please remember that thing. Library. What is the meaning of library? Library is nothing but whatever your DNA fragment attached to the adapter along with your barcode. Because adapter containing 3 segments. One is complementary sequences, 2 your flow cell and barcode and binding site. Yes, that is called library. Then we are amplifying, yes, where? A water drop in oil. Yes, that's why it is known as emulsion PCR. So all your DNA molecules are synthesized on the beads in the water droplet. Please remember that thing. So once one bead is ready, yeah, it will keep that thing deposited in the particular well. One well, one bead. This way we are doing in emulsion PCR. So your Roche 454, solid, everywhere you are using emulsion PCR. Yeah. Next slide, please. See, now we are coming to that sequencing and imaging technology. That is very, very important part. Because clonal PCR, emulsion PCR and bridge PCR. In the bridge PCR, that is Illumina, we are using chain reversible termination. That is known as CRT. So in that thing, what it contains is also nothing but nucleotide, same DDNTP, dideoxy-NTP is there. That's all. So whenever, yes, each one, that's why it is known as one thing it is find out, then you are terminating that part. So one cycle containing 4 nucleobases, 4 fluorophore, different color will go and then attach and it will read. It will take the image. Yeah, it will. We are taking the image. Please remember the next slide, please. Correct. This is happening in your bridge PCR. But whenever you are coming to that sequencing by ligation, you see, this is another imaging technology. Here we are not using, yes, your DDNTP. Here what you are using, DNA ligase enzyme. DNA ligase enzyme, yes. Every time it will detect your whatever nucleotide that is present in the that particular position of your DNA sequence. So that's why primer 1, primer 2, primer 3, primer 4. Like that, yes, everything will go like that. That's why here we are giving one tabular column. See here, what base it is. So sequencing by ligation means DNA ligase identifying the nucleotides. Please remember this point. Reversible terminator technology means, yes, DDNTP is there along with fluorotax. Yeah, one cycle, 4 fluorophores along with that sequence, it will go. Then after that, it will detach. Then again, it will form new one. That is called reversible terminator. That's why we're telling sequencing by synthesis. That is why reversible dye-terminator technology is otherwise known as sequencing by synthesis. But this one is sequencing by ligation. Every time we are synthesizing, then we are terminating. Synthesize, terminate. Synthesize, terminate. That technology is there in Illumina. But sequencing by ligation means here DNA ligase identifying the nucleotides present in the that particular DNA sequence. Next slide, please. But another thing is pyrosequencing. Yes, whenever you are using pyrosequencing, here we are using bioluminescent method. So here two enzymes are playing a major role. One is A-pyrase, another is sulfurylase. A-pyrase and sulfurylase. So what is the role of that A-pyrase? Yes, DNTP becomes DNDP and DNNP. Yes. Then after that, otherwise ATP converted into ADP. Yes, this is the role of A-pyrase. But whereas sulfurylase, what it is doing? Yes, once in the presence of your luciferase enzyme, yes, already it is containing luciferin. Luciferin is converted into oxyluciferin in the presence of luciferase. So that way, whatever ATP generated, yes, it will form the light. So that is detected by the machine. See, that is called pyrosequencing. Please remember the thing. Pyrosequencing, sequencing by ligation, sequencing by synthesis. Three technology is there. How we are capturing the image. Three things: sequencing by synthesis, sequencing by ligation, sequencing by bioluminescence. That is called pyrosequence. Next slide, please. But whenever you are coming to, fourth one is single molecule real-time sequencing. Yes, that's why it is known as SMRT. So here, how we are detecting that our nucleotides? Yes, here very important point is they are telling ZMW, that is zero mode waveguide DNA. So at the bottom of this thing, they are placing this ZMW with a single molecule of your DNA. Yeah, it is there. Along with your DNA polymerase enzyme. So your DNA polymerase enzyme, yes, detecting that particular fluorescent tagged nucleotide. Please remember that thing. That's why I see here fluorescent dye is there. So that particular color is there. Then automatically we will read G, T, A, G, C, C, like that we are reading. This is called single molecule real time. See, whenever you are telling real time, what is the meaning of real time? You are quantifying. Real time means what? Quantifying. So here what we are doing here, yes, zero mode wavelength DNA is there, where it is affixed with your DNA polymerase enzyme. Only with a single molecule of DNA. Please remember that thing. DNA polymerase enzyme plus ZMW with a single DNA molecule. That's why it is known as single molecule real-time sequencing. Yes, these are all your Heliscope or PacBio. They are using this one. Because you go to US, yes, most of the lab, they are using PacBio only. Or Heliscope. But you go to Europe, mostly yes, your Illumina part. But in India, very few laboratories, they are using PacBio. But because because it's very costly. Single molecule real-time sequencing, very costly. So that's why premier institutions, they are IIT, ISR, Delhi University, JNU, they are using. But the normal, yes, secondary university, this thing, all we are using Illumina part of, because it's very cheap and cheaper when compared to other NGS platform. Next slide, please. So now you know about all that NGS technology, what kind of platforms are there, what is their principle, what kind of amplification they are doing, how they are doing library preparation, and what kind of imaging technology they are following. Clearly, you know about that basic principle about all that sequencing platforms. Next. So now if you are coming to that solid phase amplification, Illumina, because this already we have discussed. Next slide, please. Yes. That's why. So here, application, if you are seeing there, yeah, one is normally we are doing sequencing. Whether it is RNA, DNA, yes. And if you want to study about gene regulation, yes. Then very important point is that single nucleotide polymorphism, especially cancer. Yes. Whole cancer research totally based upon, yes, that's why they are calling variant calling, variant calling. So variant calling is nothing but a single nucleotide polymorphism, what it is there. Then cytogenetic analysis, chip sequencing. So these are all the applications are tremendous, wide. Next slide, please. Roach pyrosequencing, yes, we are already discussed in detail. So it is a bead, your DNA fragment, primer, everything is there. One bead, one cell. Here in oil droplet, our amplification is going on. That's why it is known as emulsion PCR. We are imaging technology is pyrosequencing. With the help of bioluminescence, we are using two enzymes, A-pyrase and another one more enzyme. We are using that one. Next. Okay, next slide, please. Yes. Then ion torrent. Yeah, whenever you are using ion torrent, because this is all obsolete thing in ion torrent, because ion torrent based upon hydrogen ion concentration. So they are leading only the proton part. Okay. But this is based upon your pH change. Through pore, they are your DNA, whatever nucleotides are there, it is flowing. So there, what kind of pH change is there? So accordingly, based upon the pH change, your nucleobases, ATGC. So your pH may change. So that way your proton may change. So that way it is detecting what kind of nucleotides are there. Next slide, please. And solid sequencing, yes, already we have discussed about that part. Next. Yes. So already we have already compared what are all the platform, what kind of chemistry, what kind of image technology they are following. Everything in detailed way we have discussed. Next slide, please. So here why we are using NGS. One is less time, by which we will get more amount of data, because nowadays we are telling big data, metadata, correct? Deep sequencing. See, world is going like anything. So for that, and the cost is very low. And parallelly, you can get to so many things. At once, you can do all the things. That is the advantage of next generation sequencing. Next slide, please. Yeah. So application, if you are seeing mutation research, transcriptome research, metagenomics, DNA protein interaction, that's why chip sequencing. Chip sequencing is nothing but DNA protein interaction. And non-coding RNA or cancer, any rare disease where NGS playing a major role. Gene regulation, whole genome sequencing, exome sequencing, or chromatin packaging, any kind of, what is your interest, scientific interest. So in that we can utilize this particular technology. Next slide, please. Yeah. So here in that main part, what is the advantage of next generation sequencing? See here, very important point is you can see anything, deletion is there, mutation is there, point mutation, SNP, insertion or deletion, indels, translocations, CNV, copy number variation, fusion, gene expression. Another important point is methylation. See, that is very, very important part. That is why, see, whenever you are coming to the drug discovery, that particular drug is not working. Why? Because of that methylation status. So with respect to regarding that methylation status, very low minimum amount of research is going on. So all our culprit is methylation part, CpG island. So that's why for drug, if you want to improve the efficacy of the drug, yes, where your methylation status playing a major role. So that part you can find out in next generation sequencing part. Next slide, please.
Sir, are you not able to hear? Huh?
Here net problems are here.
Sir, are you able to see? Hello? Are you able to see, sir? Hello?
Yeah, it's going, yeah.
Hello, sir?
Yes, sir.
Sir, you able to see, sir?
No, now, yes, sir.
Yes, sir. Yes, sir.
Now, okay, sir.
Yes. And regarding, if you are coming to that data analysis, yes, normally we are using the term contigs. So contigs are nothing but contiguous. That is, it is representing consensus region of DNA. That contig we are telling. Because wherever that overlapping sequence data, because normally we are telling using the word read. Read is, yes, overlapping sequence data. That is what we are telling, contig we are telling. Because read is just information. But overlapping sequence data, we are telling contig. Next slide, please. Yeah. Then now, if you're coming to that, whenever you are doing, we are using another term, de novo, reference guided, reference. See, de novo means we are not using any reference. Without, that means it's new. Without knowing anything, we are exploring that part. So that is very, very important thing. Because whenever you are coming to the data analysis, yeah, whether for comparison, we are using a reference. But here in de novo, we are not using anything. That is very, very important part. And reference guided means we are comparing with some reference genome. So how it changes, what things are there. So that is known as reference guided. But another important point is we are telling mapping or aligning. What is the meaning of that? Because whenever you are telling reference gated, wherever match is there, that part only similar region we are identifying. But in mapping or aligning, yes, whatever that assembled sequences may not be identical to the template. So that is the basic difference between reference guided and mapping or aligning. Next slide, please. See, that's why this spread quality score, already you know about that point, because whenever you are telling fresh score quality, yes. Next slide, please. See, this is the thing. Q is equal to minus 10 log 10 P. Already you know about this thing. So that's why. Suppose you want 99.9%. That's why in all next generation sequencing, we are using 30 Fred score quality. Before that, don't do any kind of analysis. Then if your quality score is less than 30, then you trim those sequences. I'll show you that one. Next slide, please. Yes, next slide. Yeah, see, these are all peak deduction. So that way, each and every base that is deducted. Next slide, please. Yes, next slide. See, this is in our next generation sequencing. You will get three colors. One is green color, another is brown color, and pink color. So always you have to see, if you are seeing here, this is called box plot. So this is your median and the low end and higher end. So you have to, all should be within green color only. If anything goes wrong, because see, it is this green color starts with 28 Fred score. Your y-axis is Fred score, and x-axis is your position of the read base pair. How many base pair? So if you're seeing here 30 above 30, how many thing is there? It's around so only 6 reads. Up to 6th position of the read showing more than 30. That means this is very bad quality. So don't accept. That is why always you have to, because they will provide this quality score thing. You are seeing like this, don't accept. You can do, again you redo the sequencing. Yeah, always remember that thing. Otherwise, what will do? If you don't know this part, they will give as it is. Then while you are doing data analysis, you may face a lot of problem. And next slide, please, sir. See, here gene prediction, whatever this thing, already you know about that thing, based upon sensitivity and specificity. Next slide. Yeah, next slide, please. So here, another important point is, one is your quality score. Another important point is, what kind of files, because once you are getting that FASTQ file, your raw data, you will get in FASTQ file. Then after that, you have to FASTQ file, you have to trim it. Then after that, you have to convert into FASTA, BAM, SAM, all files you have to convert. These are all file formats. How you are, because these are all metadata. So you cannot, accommodate all the data. So that is why we are using the BAM and SAM file formats. Next slide, sir. Yeah, that's why I told single end read, paired end read. So always you can go for paired end read, because both the sides you have to do sequencing. So that is very, very important part. Next slide. Yes, see, this is paired end read. They will ask, because normally this sequencing part, 300 cycle sequencing kit is there. Everything is a kit. 300 sequence. So 1 into 300 base pair. Otherwise, 2 into 150 paired end. Please remember that thing. 1 into 300 base pair means only single end only they are doing. But if you are asking paired end, 2 into 150 base pair. So whenever you are going for whole genome sequencing or whole exome sequencing or targeted sequencing, you have to go for, yes, 2 into 150. Yes, that means what? 2 into 150 means 150 on the other end and another end 3, 5 dash, 3 dash, 3 dash, 5 dash. Yes, 150, 150. And so these are the, because you have to fill up the form. So there you have to be clearly, you have to mention that thing. Okay. Because if you are doing small RNA sequencing, very specific, then 1 into 50 base pair is enough. Yeah. That's why what kind of work you are doing. So that's why you should familiar with this paired end read. 1 into 300, 2 into 150, 2 into 75, 1 into 50. Yeah, you have to familiarize with all these things. Next slide. That's why I told. So that is 1 into 250, 2 into 150 or 1 into 300. That is nothing but short read, medium read and long read. See, in long read, you see 1 into 1000 base pair. Next slide. See, another important way, we will make as sequence depth in terms of 6x, 10x, 12x. They will say. So that is why I'm telling you, always go for, see, 6x and 12x. So in between 10x is there. So always you go for 10x depth. Whenever you are filling up that form, you use 10x depth. Yes, that way you will get a lot of data. Then only you can do a data analysis. Suppose if you are going for 6x, yeah, if your genome is very large, then very difficult to get information. Then you can't do any kind of analysis. So always go for 10x. Next slide, please. See, coverage. Always say coverage. So coverage, how they are calculating? One is your length of the length in nucleotides of the read. That means read length. Then number of sequence reads. So read length into number of sequence reads, the total divided by your length of the your sequence. So that way they are calculating that coverage. So please be careful about your coverage, because that's why we are normally, whenever you are doing that rarefaction, whenever you are doing data analysis, we will say rarefaction. So what is the meaning of the rarefaction? Rarefaction is nothing but your sequencing coverage only. All sequences covered. Have you done deep sequencing? That shows rarefaction. Please remember that point. Next slide. So these are all the glossary of that thing, because you have to know what is adapter, what is a barcode, what is target enrichment, what is a gene panel, like that. Next slide, please. So these are all the basic part you should know about: paired end, single end, depth, coverage. Next slide. Yeah. So that is very, very important part. Next slide, please. So now that's why I told. So give me 5 minutes time, I will finish it. So here that format is very, very important thing. Normally we are using FASTA format. Okay. FASTA. We have to get immediately. That is why it is known as FASTA file format. Next, sir. Next. See, that's why you will get FASTQ format. Please always. So FASTQ is text-based format. Please remember that thing. FASTQ is text-based. Second, sir. Next slide. See, it is always start with like this. So that's why in FASTQ file, yes, last line showing your score, first score. Just you should know, that's all. Because automatically your tool, bioinformatic tool, will take care of everything. But these are all the basic information you should know about the thing. 4 lanes are there. One is it will start with @ then your sequence, then plus, then your first score quality. So that is very, very important thing. This is your FASTQ format. How you are telling FASTA format? Greater than. Like that FASTQ format, yes. There are 4 fields are there. @ then your nucleotide raw sequence letter, then plus character, then your first score. Next, sir. Next slide. See, this is all ASCII character they are following. Previously, they have followed ASCII base 64. Nowadays, we are following ASCII base 33 table. Please remember that thing. What is the meaning of each and every ASCII character? Meaning is there. Please go through that thing. ASCII base 33 table for NGS data. Next, sir. See here. Yes. Next. Yes. Because you should know about that thing. See here, that's why you know this thing at the rate, this thing, colon, number 73, what it denotes. So first at the rate, below at the rate, it is showing what is your instrument name. That is very, very important thing. Because if you are coming to Illumina sequencing, so many things is there. High sequence is there, my sequence is there, true sequence is there. Yes. So that is why that instrument name is there first. Then after that, whatever after colon, whatever it is number is there, that is flow cell lane. Then after that, number, tile number. So lane, tile number, then your X coordinate, then Y coordinate, then your index number, and your whether it is paired end or single end. 1 means single end, 2 means paired end. So that information we should know what we are doing, whether it is because they are giving. Suppose you are telling paired end, whether they have given paired end or not. How will you check? So that's why you should know about these things. Okay. Next. Okay. Next. Yes. These are all. Yes. And another important point is all your next generation sequencing thing you have to submit to sequence read archive. SRA. Because normally Sanger sequencing we are submitting in Bankit, NCBI Bank IT, Bankit. There we are submitting. But if you want to submit your NGS data, you have to go for sequence read archive, SRA database. So you go to NCBI, type it SRA, you will get all the informations. Okay, next. Yes, next. Next is first you done. See here, that's why I told FASTA format is your sequence data, but FASTQ is your text data. Then SAM is sequence alignment map format. But BAM is, see, SAM and BAM same because this is sequence alignment map. But compressed SAM format is BAM, binary alignment map. Please remember that thing. SAM and BAM. Always you have to remember that thing. Further you are compressing. That is known as CRAM. CRAM. Compressed file format of storing biological sequences aligned to reference sequence. Then only it is known as CRAM. Please remember that thing. Because in SAM and BAM, no reference sequence is not there. But CRAM is compressed, but whatever biological sequences of your sequence compared, aligned with your reference. That is known as CRAM. Please remember that point. Next, sir. See, GFF, GTF, these two things is very, very important thing. General feature format, GFF. So there what information you will get? Description about gene, DNA, RNA, protein sequences. Description is there in GFF, general feature format. But GTF, gene transfer format, containing only gene structure. Please remember that thing. GFF is description about the gene. But structure you want to description, then GTF, gene transfer format. Okay. And SRA, they will say short read archive. So SRA, they will say short read archive. Okay, next, sir. Yeah. So now we are discussing about that SAM. Yes, sir. Go ahead, sir. Yes, go ahead. You come to last, sir. Yeah. SAM format, SAM tool. See, this is GitHub is there. See, now suppose if I don't know anything, I want to know something about next generation sequencing, what to flow, what workflow. If you want to know, then you can go for this GitHub, GitHub. You type it in Google automatically. So there are a lot of notebooks are there. So you read and you will get a lot of information. So that way you can enrich your knowledge. Next, sir. That's why I told. So good quality score means you have to have that green color. Next, sir. Next. Yes, you can go to last, sir. Yes, last. Yes. See, this is the thing. So these are all the applications of your next generation sequencing. What you are going to do, whether are you going to do DNA or RNA? Because whenever you are coming to DNA, one is genome aspect, another is epigenome aspect. Whenever you are epigenome, you want to do in DNA methylation or histone part, then that is epigenetic part. Epigenome you have to do. But your targeted sequencing, whole genome sequencing, exome sequencing, metagenome sequencing, these are all coming under genome side. Please remember that point. Then whenever you are coming to RNA, transcriptome, yes, whole transcriptome sequencing. They are also long non-coding RNA, short non-coding RNA, targeted sequencing. So many applications are there. So that's why you have to, because in Google, lot of informations are there. Even YouTube's also there. So you have to read and you can get a lot of information. Then accordingly you can, yes, because our whenever you are working in university or in lab, very important point is we have to publish our result in high impact factor journal, especially Q1 journal. Because whenever you are applying for leading research institutions like Delhi University, JNU, ICER, IISc, clearly they are mentioning Q1 journal only you have to publish. Otherwise, they will not consider your application. Then another important point is nowadays what they are advertising, your NIRF ranking. NIRF ranking below 20. Those candidates only you can apply. See? So that is why day by day stringent measures they are taking. So to improve our research quality. So we should note down all these things. Then accordingly we have to chalk out our program, research program. Don't publish here and there. Then that way it will not give any weightage. Please remember, whenever you are in that, either you are in the beginning stage or middle stage or a senior stage, everywhere now quality playing a major role. Without quality, you cannot sustain. So that's why please note all these things and publish papers in Q1 journal. Q1 journal. That is very, very important point. Then only we can improve our quality. People may say this way, that way, but this is the thumb rule we have to follow. If you want to improve your research quality, improve your lifestyle, then you have to publish only in Q1 journal. Okay. Thank you so much, sir.
Thank you, sir.
Yeah. Do you have any questions? Now you can ask.
Participants are invited to your queries. Sangeeta madam.
Very good afternoon, sir. Sir, there is a question from Dr. Guha Rajshekhar. What is the length of the reed in Sanger?
Madam,
one question from Dr. Guha. That is, what is the length of the reed in Sanger?
Madam, can you please type it in that chat box? Because your voice is
Sure,
breaking.
Sir, can I ask question directly?
Yeah, sure, sir.
Yes, sir. Sir, myself Rajshekhar, sir. I'm actually faculty. I have attended this program. Sir, what is the length of a read in the Sanger sequencing? And my other question is, if we use R programming and Linux, both give the same result or they use for different purpose? And one more thing, which is more easy, using Linux or R program for analysis of genomic data?
Yes, it's a very good question, sir. It's a basic question. You see, Sanger sequencing length is maximum 1000 base pair. That's all. And you can target only one gene. Your gene target is only one gene. Then only, yes, that's why we are telling there's a gold standard at those times. But now, yes, in NGS sequencing, you can parallelly, you can target several genes as a whole. That's why we are telling whole genome sequencing, whole exome sequencing, whole RNA, transcriptome sequencing, metagenome, whatever microbes are there, whole meta, yes. That's why we are telling metagenome sequencing. Shotgun sequencing, if you want to know the functional part of that particular microbe, anything it is producing, then we have to go for shotgun sequencing. This is question number one. And number two, see, this Linux is the platform, sir. It's a command line, because normally we are accustomed to Microsoft Word. So whenever you want to work in next generation sequencing, always don't install Windows. You have to always use Linux, that is Ubuntu. U-B-U-N-T-U. Ubuntu, yes. You have to use Ubuntu. Yes, this is Linux based platform because everything is command based. Yeah. And another important point is now this R language, R programming, yes, you can use in that Linux system. R programming is specially for data analysis. Anything you want to draw a graph, or have you seen all Q1 journal published papers you have seen? So many analyses are there. Venn diagram you want to prepare. So you can't do it in any other software tool. So for that you have to use your R programming. So that is why we should know basic thing, because already commands are there. But how to use that command? For what purpose I have to use? So for that we have to use, for example, heat map. One is heat map. Whenever you are working on metagenome or transcriptome, heat map, that way we are projecting our data. So heat map, if you want to do, yeah, then you want to use that R language commands. So that way, nice way you can present your data. And another important point is whenever you are working on metagenome, there is another software is there that is called KIME. Q-I-I-M-E, KIME 2. See, all, yes, whenever you are working on metagenome, yes, even if I don't know anything, yeah, but I want to do something, then you use KIME 2. Yes, it is very simple thing. But thing is, you have to attend one or two training programs. Yes, who is working, who is doing, you have to learn, and that's why. That's why if you know that Linux programming as well as your R language programming, then easy way you can access all the data. That is very, very important thing. But without any knowledge about your system or platform or pipeline, you are attending any workshop, no use. Because you don't know many things, no? Then how will you ask question and how will you progress further? Because see, I joined in this university in 2007. That time I didn't know ABCD of anything. After that, yes, myself, I attended so many workshops, this thing, reading. So that way I have developed all the things. So same way, same procedure. Yes. Very important part is our strength is our students. So always, yes, train our students because they are catching up is very fast. So that is why always train our students, and yes, that is very, very important part. So our duty is to train our students. Once you are trained students are there, then things will go smoothly. But you have to do a lot of hard work, because without because problem with student community is they may not pass the information, and they may not think that like us, because we are the doer. So what to do, what we have to do, that the responsibilities are there. But at students' life, yeah, you tell sir, I will do it. Like that only, that approach is there. So that's why we have to train our student. And that's why if you don't know ABCD of bioinformatic part, no problem, sir. A lot of YouTubes are there in regarding that Linux programming or R language. That's why I told you GitHub. Yeah, you type it GitHub. You type it that GitHub, there lot of repository are there. So what do you want? Yes, you can collect slowly. You have to go one by one, and yes, and I'm telling you, within 1 year you will become master. Yes, and please refer literature. So how they are projecting their result, what they want, how they are yes representing the data. Because whenever you want to publish in Q1 journal, that analysis part is very important thing. Your analysis is not good, then you cannot publish in Q1 journal. So that is why be careful about that part. I
Sir,
hope I
one
have
more
answered
question,
your
sir.
question. Yes.
Sir, one more question. It is related to Human Genome Project, sir. Sir, while they were doing Human Genome Project, what was the reference genome they took for it, and how they assembled the genome, sir?
No, that time genome, wheat genome, human genome, any genome project, all genome project, yes, all de novo only. But we have collected all the, see, the first part is, beginning is Sanger sequencing. So that way your NCBI database is there. So we have collected all the data. All, yes, you go to NCBI database, whether it is a protein or nucleotide or any kind of thing, all data are there. Metadata is there. So from there we have started, because now we have to find out what it is. So for that you want DNA barcoding needed. Without Sanger sequencing, you can't do it further. But now we have collected all the data and put up in that database. That's why a lot of reference database is there. Suppose, for example, if you are working on metagenome, Silva database is there. So in that Silva database, you show your transcript, it will identify what kind of microbes are there. So for that, you have to go for R programming. That command is there. So you put it, yes, automatically everything will come. But you have to have very good high-end system. That is number one. And you have to basic knowledge about all these things. That is very important thing. Because every day is a learning process. But still, you see, still I am learning. I don't know many things. I will ask the expert. They will tell, yeah, you do it like this. Whatever problem I am facing, yes. That is very important part. Because you cannot master all the thing. Huh? So that is why we have to collaborate with overall your frequency matching, where it is matching. So you have to get a help from them. But nowadays in YouTube, everything is there. You type it, what question, yeah, answer, everything is coming. So that way, because we are all learned persons, so we can pick up very fast. It's not a big thing, sir. That's why I don't know ABCD of anything. In 2007, yes. What is bioinformatics, platform, pipeline? I don't know ABCD of anything. After coming here, I have developed everything. Yes. Only yes. Our vision and mission. What is our vision and mission? If you want to achieve, you have to achieve. Yes. Because though in Mizoram is a remote area, nothing was there. Yes. With the help of the Department of Biotechnology, DST, DBT, ICMR, we have collected funds and developed all the things. Yes. This way only you have to proceed further. And especially if it is a private part, a lot of difficulties are there. No one will give any support. But actually speaking, private universities are doing well than your government universities. So we have to overcome certain things. But if you are attending a workshop related to your field, then you can gain a lot of information. Yeah, no doubt about that, sir. Yes.
Thank you so much, sir.
Like that
Thank
only.
you.
Thank you, sir. Thank you, sir.
Sangeeta madam.
Sir, one more question. Can we analyze mitochondrial DNA sequence by NGS?
No, my point is, madam, your previous Dr. Gupta Rajashekara, G. Rajashekar, he asked genome project, all genome project related to nuclear DNA only.
Okay.
But apart from that, Apart from that, mitochondrial DNA is there. Your chloroplast DNA is there. See, especially our group is working on mitochondrial DNA. See, I am working on insect. So for DNA barcode, I have done everything in mitochondrial DNA. Yes, madam, please. Yes. What is your question?
Can we analyze mitochondrial DNA sequencing by NGS?
Or
Sir, in a chat box, I have some more question.
no, yeah. Can we analyze mitochondrial DNA sequencing by NGS for forensic application? Yes. Madam, please remember that thing, because here, that's why in isolation step, extraction step, if you want mitochondrial DNA, little bit, two more steps are there. You have to increase that RPG, that is X. You have to do that because you are going for organelle type. Because you have to separate it out all mitochondria. But normal thing, you are separating only nuclear DNA. So only that separation step, little bit differences are there. That also depending upon your microbe or plant or animals, that will differ. So you please refer literature, then accordingly you can do that thing. Fantastic. Yes.
Thank you, sir.
Yeah. That's why you please refer what published paper is there with respect to your mitochondrial DNA. What kind of papers are there? Because without paper, we can't do it anything. That is very, very, because we are not experts, no? We are learning. So that is why we are referring the literature. Accordingly, yes, what they have done, just we are repeating in our whatever little bit, you are changing the modification. Because in forensic thing, yes, lot of thing there you have to do. So many things are there. But still, because the government, they are following only that recombinant DNA technology, old thing only they are following. But after that, NGS, whether really they have applied into forensic science or not, I think you check with PubMed. Any papers are there or not?
Okay, thank you so much, sir.
Because problem is, we are doing basic research. In basic research, you can compile anything. But your part is professional, forensic application. So whenever you are coming to forensic application, yes, really, whether whatever data it is there, whether is it useful, immediately you have to utilize that thing in practice. So definitely lot of scope is there with respect to NGS. Yeah. No doubt about that.
Thank you. So there is no more questions, sir. I would like to conclude this webinar. I, Dr. Sangeeta Patle, on behalf of Department of Forensic Science, Mangalayatan University, I would like to propose heartfelt thanks to our guest speaker, respected Professor G. Gurusubramanian, for gracing today's webinar on next generation sequencing and its applications. Thank you, sir, for sharing knowledge with us. The insightful presentation was truly thought-provoking, and I believe we all have gained valuable insights. Thank you, sir. I would like to express my profound gratitude to our Honorable Vice Chancellor, sir, K.R.S. Sambashiva Rao, sir, for his presence in this webinar. Thanks to all professors and colleagues and all the participants. Once again, I thank you one and all. Thank you.
Thank you. Thank you. Thank you so much.
Thank you, Gurusubramanian.
Thank you so much, sir.
Thank you. So
Thank you
very
so
much. Thank you very much.
Thanks. Thanks.